you very much, Mike, and good evening, ladies and gentlemen. I'm very pleased to have the opportunity to present to you about AGL PLC's liquid biopsy technology. I believe this technology has the potential to transform cancer care. I think it potentially offers everybody here a very interesting healthcare investment opportunity. However, I know that not everybody here has come for healthcare because we've had 3 very good presentations on other areas. Nevertheless, I'm sorry to say that this presentation is probably relevant to everybody in this room because Cancer Research UK now tells us that 1 in 2 of us will suffer from cancer in our lifetime. So there's a 50% chance that any one of us will get cancer, and there's a 50% chance that any one of our loved ones will get cancer. I'm sorry that that's the case, and it is, of course, very, very important to try and find ways, and the industry is trying to find ways to improve the care for these cancer patients in the future. And AGL PLC is involved in the vanguard of the latest efforts for personalised cancer care. And what that means is providing care that meets the specific cancer requirements of that individual patient at that individual time. And that is something that is extremely difficult to do. What we have in AGL PLC is a system, a technology which is fully patented. It's a product which can enable a simple blood test to help with personalised cancer care. This has been proven out now with multiple different cancer centres in Europe and the United States, and is addressing a very large emerging multi-billion-dollar market. The one thing I would suggest you take away from this presentation is that the area of liquid biopsy, which I'm talking about, is definitely coming. The question for all of us is to the extent to which AGL is one of the leaders as that market emerges. But you can see there that Goldman Sachs, I think the market in the United States alone is going to be over $14 billion in the future. We believe AGL has the potential to be a leader in that marketplace. In terms of our operations at the moment, what you can see from our financials is we are already making research use sales in this market. We consider those levels of sales to be quite modest, but they are nevertheless very important because leading cancer research centres around the world are buying our product and they're using it to do research and development into new potential applications. There are over 20 different cancer types being investigated by these customers and by our key opinion leaders using our Parsortix system. The other important thing on this slide is the level of investment that the company has been sustaining with the support of our shareholders and their investment in our company. And the reason we're doing that is because we're leading many leading clinical studies in different cancer areas. Because if you want to have a product utilized in a healthcare market, it needs to be proven. You need to have clinical studies with patients showing how their patients will benefit from whatever the area is. And it needs regulatory clearance. So the government agencies involved in the different countries need to give approval for a product to be sold. And that is a very, very challenging environment in which to get success. And that is why our company has been investing consistently to achieve that. And in terms of our cash position, at the year end, the 30th of April, we had £5.5 million. But subsequent to that, just this month, we've completed a £15 million fundraise. So we're now in a strong position to execute our business plan. And the other thing to note, that in the research use sales that we have achieved, we get high margins. We have a great product which we're able to sell at a very, very good margin. So in terms of the whole area, why is liquid biopsy needed? Well, the answer is that each patient's cancer is different. We imagine that 3 men who have prostate cancer have the same disease, but in fact they have the disease in the same part of their body. Same thing with breast cancer. 3 women with breast cancer will have different diseases, but in the same part of their body. And the problem and the reason that we have high mortality is that the cancer treatment is not targeted enough. It doesn't take into account the fact that these cancers are different. And what's more, they change over time, so that a cancer may present in a certain way initially, but it mutates and changes during the patient's disease. And that's why we hear of great progress being made in the treatment of an individual's cancer and the reduction of the size of their tumor, only to find out later that they've relapsed and it's spread and it's uncontrollable. The reason it's uncontrollable is because the doctors don't know how to treat it. And the reason they don't know how to treat it is that they're basing their decisions on solid tumor biopsy. So that means cutting some of the cancer out so it can be analysed. If it's breast cancer, the lump is cut out or a mastectomy is done. If it's a prostate cancer, it may be a biopsy. You look at the cancer cells that are cut out, and from that make the decisions about treatment. But if it changes, what can you do when you've already cut the lump out? So the woman's had a mastectomy, she's still got breast cancer, it's changed its spots, it's not responding to treatment anymore. How do they know how to treat the woman going forward? The answer is it's trial and error. And this is the same with all the different solid tumour types. However, if you could have a different approach, which we're pioneering, a liquid biopsy based on blood, then you could just do a simple blood test whenever you want and check what is the current treatment that this patient needs. So why can you do that? The reason you can do that is because inside the blood there will be some cancer cells. So if the woman has breast cancer, then that first tumour, the primary tumour, is shedding cells into the bloodstream, cancer cells which circulate, and they're known as circulating tumour cells. And these cells are the most important cancer cells that exist because they're the ones that land somewhere else and cause a secondary cancer. And we all know that over 90% of people who die from cancer don't die from their primary, they die from their secondary. How did the secondary cancer go from the breast to the lungs or to the bone? It travelled in the blood, and it was a circulating tumour cell. So if you could get that cell and look into what's driving that cell and find therapies to address it, that's how you could make a difference to the outcome for the patient, which of course is what we all want. And the advantage of a blood test is it's non-invasive, it's repeatable, it's real-time, and it's cost-effective. So what's not to like? Well, the answer is it's incredibly difficult to get hold of the cancer cells. There's only, on average, maybe one cancer cell in 1,000 million blood cells. So it's technically a phenomenally difficult challenge. That is the challenge which our company has solved with our Parsortix technology. And what I have here in my hand is a Parsortix cassette. And afterwards, if anybody's interested, I can show you these, and so can Ian Griffiths, our CFO over here. This is a very simple piece of polycarbonate plastic with a thin film of plastic on top of it, which makes a void inside it. And we flow blood inside this cassette. It has some patented steps. So this is a product which we own worldwide. And these patented steps are plus or minus 1 micron, so it's very, very precisely manufactured. And they are set at a level where the blood cells flow through, but the cancer cells, which are larger and less compressible, get held inside the cassette. So it's an incredibly simple process. It doesn't kill the cells, but it does enable them to be harvested. And so this is what it looks like. So the blood comes in through the inlet. It flows down a channel and it goes out of the outlet, so it's inside the cassette. And if you look at this edge here, what you can see is a series of steps, so those are the patented steps. And this is an illustration showing the smaller red blood cells and the white blood cells streaming up the steps through the critical gap and into the outlet trench, whereas the larger cancer cells shown here as green are held gently in position. There's no damage to these cells. 99% of them come out alive, so they express all of the proteins, RNA and DNA needed to investigate the cancer. So that was an animation. This is a real-life example at 100 times magnification, and what you can see is red blood cells here streaming up the steps, so that's looking down on top of the staircase. Through the critical gap, there's only a single cell in depth here. The main point about this is it flows very easily. It's a very simple process. Basically, we're using a piece of plastic. And here's the blood in the outlet trench. And what happens is if you find a captured cell, so that is a cancer cell inside that. So remember, there's thousands of millions of blood cells and just a few cancer cells held gently on the final step. They're always in that location, and the rest of the blood flows around it. So this is a very, very simple engineering solution to an incredibly difficult biological and molecular chemistry question. People have been working for years and years and years to try and crack this problem. It's just very, very lucky that a microfluidic expert in Philadelphia came up with this idea, patented it, and then Angle invested and built a company around him quite a number of years ago. So, just to summarize, what is it that we have in terms of our Parsortix technology? Well, we focus on intact circulating tumor cells. These are living cells, and the relevance of this is that the competition are focused on the thing next to it, the ctDNA, which is fragments of dead cancer cells. Problem with that is when a cell dies, it goes in the bloodstream for about an hour before it's excreted, but it's split into millions and hundreds of millions of little fragments, so you can only look at a partial bit of the picture. You can only see some DNA, you can't see any RNA or protein expression. We capture living cancer cells, and the real relevance of that is you cannot have a living cancer cell in your bloodstream unless you have cancer. And that's what we call specificity. The technical term is specificity. So when we detect that, we know the patient's got cancer. With this technique, and actually worse with most normal techniques like protein expression, so many of the men in this room may be aware of the PSA level, which is a blood test that's taken to assess whether the presence of prostate cancer. That PSA level is a protein expression. It's not a living cell, and it can be upregulated for lots of reasons, nothing to do with cancer. So if you have a high PSA level, don't assume you've got cancer. You've got an 80% chance you don't have cancer. We're driving the process here with key opinion leaders and clinical studies I mentioned earlier. It's a patented solution. This is an instrument that we've developed and own. It automates the whole process. So a simple blood draw, just a normal tube of blood, is put on the middle of this instrument, and the user pushes start, and it just automatically pushes the blood very gently through the cassette, captures the cancer cells, and washes away the remnants of the blood into the waste container. So it's a very simple process. Those of you who, I know some of you in the room follow Angle, and those of you who follow Angle will have seen that we've just made an acquisition, and that acquisition is the assets of a Canadian company called AGL Inc. Now, we use this company to do some very clever analysis for us in our ovarian cancer study, and it proved that their technique was extremely sensitive at analysing the cancer cells that come out. So the long and short of it is we now own also, as well as the cancer cell capture, this is all new information for you that have been following us. We own the AGL assets, and that means that we can analyse the cells that come out of the Parsortix system. What's great about that is it means we now can offer the full solution, all the way from the blood draw through our process, and out can come the gene expression information. And in our ovarian cancer detection study, they looked at over 60 different gene expressions for us and showed very high sensitivity and specificity. I'll jump over this, but it's highlighting, and you can see this in the PDF, which is going to be sent around, but this is highlighting the advantages of the intact cancer cell, the circulating tumor cell, in terms of its ability to do things, DNA, RNA, and protein, compared to the competition, which is ctDNA, where it can't do RNA and protein. And obviously also compared, here's very interesting, this is trying to do a biopsy, a solid biopsy of a metastatic site. So in breast cancer, for example, in the United States, if possible, the woman will have an operation to have a biopsy of her secondary cancer site. And if she's got cancer in her liver, that means a general anesthetic and a slice of her liver is cut out. We've had, I'm going to jump the slide when we get to it, but we've had a study that shows you can get the same information from a simple blood test using Parsortix. So if you've got this groundbreaking technology, which can get cancer cells out of the blood of cancer patients, enable them to be analysed to change the way these patients are treated, what can you do with it? Well, the answer is a huge number of things, all the way from early detection of cancer, because these cells shouldn't be in the bloodstream, and if they are, then the patient's got cancer. Even if they've got no symptoms, you can detect that, and that's what we're doing with our ovarian cancer study, through to selecting the right drugs. I find it completely shocking that the new drugs that come through, which are phenomenally expensive, up to £150,000 per year per patient for a new immunotherapy. Those immunotherapies are great because if they work, they can stimulate the body's immune system to reduce the cancer down dramatically. Problem is they only work for 1 in 4 patients. So you've got 3 in 4 patients, £150,000 wasted. And not only that, the patients have got toxic side effects from the drugs, and they're not being treated with drugs which will help them. So wouldn't it be better to have a blood test to work out which patients should have which drugs? And that's where we're focused. And then there's a question of patients who have finished treatment, have they still got any disease remaining? And patients, this is an absolutely huge area. Patients who've had cancer, they're in remission. Obviously, what do you worry about? The worry is a possible relapse. You can find that patients have a relapse 10, 15, even 20 years after their primary. How does that happen? The answer is that these circulating tumor cells, the cancer cells I've been talking about, hide away. They hibernate in the bone marrow, and then something causes them to come back out. They're called disseminated tumor cells when they're in the bone marrow, but it's basically the same thing. They come out and they can start a new cancer, and by that time, usually, it's too late to treat the patient by the time they've got symptoms. So wouldn't it be better to use Parsortix and actually look and check, say, once every 6 months, there are none of these cells circulating in the bloodstream? So the long and short of that is there's any number of different applications for this technology, and we want it to be used for all of them, but we're a small company. So what do? We've got a very focused approach where we're working on ovarian cancer and metastatic breast cancer with some work on prostate, which we're doing to prove out the access to those specific markets. So they will open up major markets for us. The ovarian cancer one, for example, is worth £300 million a year. We've already had 2 studies which show that we outperform standard of care dramatically in detecting ovarian cancer. So the idea is that these will open up clinical markets to Angle, but also will prove the exemplar which gets the large companies interested in using our technology. So why would the large companies want our technology? Answer, they've got an installed base of very, very high-powered molecular testing equipment already in hospital labs. But it can't do liquid biopsy because they can't get the cancer cells. They can do solid biopsy, so they're being used for solid biopsy. But if we can give them the ability to get hold of the cancer cells, then suddenly they can use their install base and get much larger levels of sales. So we're following a process on the development of our individual applications. Ovarian cancer has been through 2 large-scale verification studies, both of them very successful, one in Europe, one in the United States, 200 patients each. The metastatic breast cancer, this is an absolutely key area for us. We're focusing in on metastatic breast cancer as our first FDA clearance. So what does that mean? Okay, so the FDA is the US regulatory clearance system. You cannot sell anything for treating patients in the United States unless the FDA blesses your product, and you have to do all kinds of very difficult analytical and clinical studies to achieve that. It's also deemed to be the gold standard for the rest of the world. So even here in Europe, everybody looks to see, have you got FDA clearance? In this space, which is a multi-billion-dollar market coming through, there is no company that has a circulating tumor cell harvesting FDA clearance. We're hoping to be the first ever. And we have a study which is a validation study due to complete end of June. I'm going to jump over some of this, but the most important thing here in the ovarian cancer is the fact that we had very high sensitivity and specificity. So the best tests currently available in detecting ovarian cancer have approximately 5 false positives for every true positive. We hope to reduce that to less than 1 false positive, which would be a major change. Large market, 750,000 women a year who have an abnormal pelvic mass that would need this kind of thing, and that's in the United States alone. This is the breast cancer commentary. So basically, we've got 200 metastatic breast cancer patients and 200 age-related healthy volunteers. And we have to prove that our system successfully gets cancer cells from the blood of the metastatic breast cancer patients. MD Anderson, that's the leading cancer center in the whole of the United States, has taken on doing that work for us. And we have 5 other leading US cancer centers. Key point is that the results of the clinical study and the analytical studies are due in H1 2018. So we're getting very close to prime time with Angle. This is some work I mentioned earlier about the difficulties of biopsying the metastatic secondary cancer site. And this lady here, University of Southern California, Julie Lang, has shown the same results coming through with the Parsortix blood test compared to the metastatic biopsy. I won't dwell on this, but I mentioned prostate cancer and how important that is. Difficult thing with prostate cancer is A, detection, because the PSA level is very non-specific, but more importantly is, has the patient got an aggressive cancer or not? Because half of all prostate cancers are likely to be fairly low level and very slow progressing. And many men in this room will die with prostate cancer with not even knowing that they've got it. So it can be a very low level thing, doesn't provide any symptoms. Alternatively, it can be very aggressive. What this chap did, Yonchi Liu at Bart's Cancer Institute, was run a 40-patient study using Parsortix, separated the 40 patients into a high-risk group and a low-risk group, and hey presto, the high-risk group were 10 times more likely to die in follow-up than the low-risk group. Obviously, it's very important to know which group the patient's in order to make sure they get the right treatment. I'm going to just wrap up now. This is very important. I mentioned about our strategy to partner with large companies to get rollout. This QIAGEN is one of the world's leading molecular testing company, $1.3 billion of revenue in molecular testing. They have all kinds of downstream analysis techniques. They've chosen to use our Angle Parsortix system as their front end for that, and we signed a co-marketing agreement with them about a month ago. By the way, we signed one this month with Philips, who have 17 billion euros in healthcare, and they want to get into I'm going to leave this, actually. Basically, audits took... Do you want to come and join me, Ian? So this is talking about the AGL system, and the two systems together, we believe we can do a better job than what's already here, and they had $300 million of revenues last year in solid biopsy. So perhaps if we can take some questions.
Yeah, just down at the front. You said you had a patent. Is that correct?
Yes, we do. We have several patents.
What is the dating on those? Are they going to run out in 10 years or 5 years? Or?
So there's a range from sort of 2023 up to 2035.
And is there do you think there's any chance of someone else being able to obviate those patents? Or?
Not to obviate the patents. I mean, that's the whole point when you get a patent, and these are in the major markets in US and Europe. And obviously, for us, we've also got patents out in China and places like that as well. So, it doesn't mean there won't be a major company that will try and challenge it or do something, but we've tried to cover the major markets. We've had freedom to operate, so we're confident in the ability to, protect our position with those patents.
Are there any types of cancer your company's product can't detect?
We're not working in blood cancers because leukemia and the like, they have a lot of cancer cells in the blood, and so the technical challenge of getting hold of those cells is not so relevant. But as I mentioned, our customers and KOLs are working with 20 different cancer types of solid tumour. And is it about 90% of cancers are solid tumours?
Yeah.
I mean,
every cancer that either we, our key opinion leaders, or our customers, every solid tumour cancer that has been tried, they've got CTCs, so we have no reason to believe it won't work with all solid tumour types. Again, it's a reflection of the fact that the primary tumour is shedding CTCs that circulate around the blood, so we capture those CTCs.
There's a large number of independent cancer centres who've written peer-reviewed publications, and those are available on our website. Most recently today, there was one from Medical University of Vienna talking about the efficacy of the system in ovarian cancer.
And I think probably one of the things I would add, that has historically been a bit of a problem for CTC companies because some of the technology they used was antibody-based, and that only works with certain types of cancers that express certain characteristic. We work with all CTCs, so we can work with all cancers as a consequence.
Just there.
Hi, Andrew. How far are we from the FDA approval? And, what is your opinion about, we have a lot of positive RNSs being announced one after the other in the last few months even. And, the share price always tend to retrace for some reason.
So there's two different questions in there. I'll start with the FDA clearance. So we've been working, as you know, at this for several years, in fact. The challenge of actually designing the study to meet all the FDA requirements, that's taken well over a year of intensive work with the cancer centres. We hope to have the first patients enrolled in well, possibly late December, but more likely in January. And after that, we've got 6 cancer centres who are all actively recruiting and enrolling patients. So we're reasonably confident that they can complete the clinical study by the end of June next year. In parallel with that, we've been working for quite a long time on the analytical study to do with the reproducibility and the robustness of the instrumentation, and we're confident that will be completed. But the nub of your question was actually, when will the FDA give us clearance? That's a different question because it's not in our control. What we anticipate is they will come back with a series of questions. Now, we've been working intensively with them as well, hopefully trying to meet these questions, but they may come back with questions which we then have to answer. So there's likely to be a process. We would anticipate towards the end of next year would be about the earliest time that we'd actually get the clearance. We think there'll be very, very active interest in the company after the studies are announced that they've completed.
Would that clearance allow you just... Sorry.
Oh, the share price question. Do you want to try that one?
Yeah. So, I mean, it has been somewhat volatile. There's been, I think, various factors. So when we put out the ovarian cancer results from the validation study, which we thought, was our biggest piece of news, the market didn't respond. And some of the feedback at the time was people didn't realise we needed to do more studies after that. They thought we'd be able to go straight to market, even though we'd flagged at previous presentations in our annual report and account. There's a process we have to follow, as Andrew put up on the slide. That was a validation study, and then you've got to do your verification study. And so we're in a period of optimization now. So I think some people thought, oh, it's taking longer, and they hadn't realised there's going to be other studies, and that got some negative sentiment. We did have one of our major shareholders was changed ownership, and they had, for technical reasons, had to sell out quite a chunk of their shares. And so that put some downward pressure on the price as well. I think there's just been that little bit of negative sentiment. So we did go down to about 32. We're back up to about 46.5 at the moment. And what you will have seen is that's been sort of more sentiment with the retail investors. The fundraise we've just completed, very, very strong support from the institutions. So hence we went out to raise 12 million and we closed on 15 million.
Next question.
You're portraying an extremely attractive company. Aren't some of the big biotechs going to want to gobble you up?
I think there's a history of the large companies waiting until things are absolutely finished, and they'd rather spend 10 times as much money for something that's absolutely finished. At the moment, we're still investing heavily, so we would be dilutive to earnings rather than accretive. So that's one aspect. But what I'm hoping is we will be successful in getting multiple companies doing deals with us with particular different cancer types or different applications, so that we have, if and when they do come for us, that there's a bit of a competitive streak. That's the aim, and that's why we're pleased that we signed now Philips as well as QIAGEN. So ideally, we're going to have dozens, well, maybe not dozens, but a lot of different companies signed up with us as well in different areas.
Well, 10 times the price sounds like a very good idea.
That was just next door to you, actually.
This is just a simple clarification. I can remember reading some medical journal and found that about 10 years ago, 1 in 3 of us will get cancer in our lifetime, whereas you mentioned today 1 in 2 will get cancer.
I was careful to attribute that to Cancer Research UK, by the way.
All right, yeah. Okay. So I believe your method of, or this technology you are introducing probably will go back from 1 in 2 to 1 in 3.
No, I don't think it necessarily will. I mean, I'm not an expert on the causes of cancer, but the reading that I've done, there's quite a lot of environmental reasons for cancer. So everything from smoking to alcohol to exposure to the sun to obesity to eating the wrong processed foods. There's all these different things. And certainly there's mega data which suggests that developing countries, as they become economically stronger, unfortunately their cancer rate goes up.
And you have to align this also to the fact that people are living longer. And a lot of cancer, it's a disease of age. Yeah. So all the improvements we get elsewhere, well, people have got to die of something, and so incidence of cancer and other sort of diseases are going to go up. But as I said, what you need to do is try and understand it better. I mean, the prostate is a classic one, because a lot of that is, what we call indolent, or it's not aggressive, so you can live with it, it won't kill you. But if you have a prostatectomy, it has a whole host of implications. And at the moment, the treatment path is to be, well, because they don't really know, they tend to be a bit more aggressive than they need to, and they will go down that procedure rather than waiting, because they can't tell you, when it's going to flip. And you get this in lots of different cancers. So you've got people with the BRCA2 gene, which is the Angelina Jolie position. So she's had a double mastectomy, she's had her ovaries removed. She's got a risk of cancer. It's a much higher one, it might be 50%, right? But she didn't necessarily have cancer at the time. Wouldn't it be better, to go through your years, and then as you have a test that can say, well, actually, we can see that cancer is beginning to develop, then you take the action. Some people are taking action very, very early, and if it's a 50% chance, then actually a lot of people are taking actions when they wouldn't even get cancer.
Sure. Okay. Thank you.
Thank you, Andrew. Thank you very much indeed. Thank you.
Okay.
Thank you.